And welcome to our final lecture in immunity. We'll go into immunizations in the next video or two, but this will be the last video in our innate and adaptive immunity lecture series. And here we'll go mostly over B lymphocytes and humoral immunity. Remember, humoral immunity is categorized by fluids and the four humors from Greek. Okay, and remember we kind of already went through this, and this is going to develop into antibodies. And those antibodies we can measure, right, in the blood, and we can see levels of antibodies in the blood, as in the case with Olivia in our case study in the beginning. Okay, so hopefully you saw that video. And a B cell receptor looks a lot like a antibody, right? And we have the heavy chain here and the variable region here, and they are embedded into the membranes of B cells. Okay, this is not quite an antibody. Remember, antibodies are more free-flowing, but it does allow antigens to bind here, just like the antibodies. Okay, so those B cells receptors will encounter a bacterial pathogen, okay? And those B cell receptors then will proliferate, and they will be uh, um, PAMPs, okay? These pathogens have PAMPs. PAMPs is a big long word of pathogen associated molecular pattern. We've kind of talked about that, danced around it when we've talked about motifs and epitopes and flagellin and lipoprotein. Okay, those would be um, uh, molecular patterns that are associated with pathogens. Okay, so PAMPs. Okay, so this B cell will detect the pathogen through the PAMPs, not detecting the whole pathogen, and then through activation of the B cell, we will activate and secrete our antibodies, in this case, pentameric IgM. Okay? Okay, and we can do that uh, independent of T cells. Okay, and then in order to do that independent of T cells, we need to have those B cell receptors that directly encounter the pathogen. In T cell dependent activation, uh, we are going to rely on the T cell that's been activated. Okay, that T cell, called the helper T cell that's been activated, will encounter the B cell. Okay, the B cell has already encountered the pathogen and undergoing clonal selection, and they kind of help each other. Okay, and then we'll create memory B cells. Okay, and then some will create plasma cells, which will release antibodies. Okay, so the memory B cells can then go and remember uh, and encounter a PAMP later on. Okay, and then go into T cell independent activation. Okay, and then or we can go and make plasma cells, like we talked about in the first slide, and secrete antibodies. Okay, so we have a few options with T cells. We can have, well, with B cells, we can have T cell independent activation and T cell dependent activation, but at the end of the day, B cells are humoral immunity in fluids that release antibodies. Okay. Okay, and then to drive home the whole point of our secondary response, okay, is that we have antibodies. Now we can detect antibodies in a person, and we can actually see how they're responding to a pathogen. Okay, so at the initial exposure, there should be no antibodies for that pathogen. Okay, but after 10 days, IgM is produced. Okay, and IgG is produced as well. Okay, they're both produced at basal levels, but in order to increase, IgG is a little slower. Okay, it's a little slower, but it's also higher. Okay, all right, and so that's our primary response. IgM, the pentamer, starts to detect the pathogen, and IgG tags along and helps finish the job through opsonization, neutralization, glutination, all of the above. Okay, then we're exposed again, and this time it's not 10 days to start or 15 days to start. Instead, it's 5 days or 4 days. Okay, and note IgM is roughly the same. Okay, in fact, it's probably actually a little lower. All right, but IgG is about double. Okay, there's about twice as much IgG in a secondary response as a primary response. Okay, and that is because it is adaptive. We've adapted to it. We recognize you. We have some B cells. We have some memory T cells, and we have some memory B cells. And if we encounter it again recently enough, we'll be ready, and we will encounter it with some more of our immunoglobulins. All right, that'll conclude our adaptive immunity and our innate immunity lectures. And I'm going to go into immunizations next, followed by immune disorders.